Hello, good morning, everyone. It's a great day today. One more day. Um, I'm really happy to have my colleague and friend, Dr. Ruth Keri, joining us today to give a talk. Ruth is professor and vice chair of the Department of Pharmacology and the associate director of basic research for the Case Comprehensive Cancer Center in Cleveland. And her research is focused on the genomic and signaling mechanisms that control mammary gland development and breast cancer. She is a rare individual. She is one of those people who have started her career at Case. She grew up in rural Pennsylvania and attended Edinburgh University of Pennsylvania with a degree in chemistry. And then she was briefly a research assistant at Case before entering grad school there. And she also postdoc there and has risen all the way up to vice chair of the department and associate director of basic research at the Cancer Center. And I think that is a testament to her just outstanding science and leadership, as well as, you know, the ability to rise to that level within your institution is pretty difficult, we all know. She's done a ton of things, not only through her research, which has gone from working in the pituitary, working in the ovary with tough endocrinological questions, but moving in the last 20 years or so into understanding mechanisms that drive breast cancer with regard to hormone action and signal transduction. She's done a ton of governance and leadership nationally. She's on the board of directors of the Endocrine Society, which I don't know if all of you know, this is a society of nearly 20,000 members, and these board of director positions are nominated and elected by your peers. So it's quite an honor to be on the board of directors there. She's had a number of governance and advisory positions at the NIH as well as FASIB. And she's led and organized a number of first-time meetings and workshops in her field, National Toxicology Program at the NIEHS, Society for the Study of Reproduction. I mean, just numerous credits to her mentorship, her leadership, and her science. And so I'm so thrilled to have her. She's been a fabulous colleague. I have to say, I wouldn't have the R01 I have if Ruth hadn't torn it apart for me. And I had this little come to Jesus, only it was a come to Ruth talk moment with her when she said, it's too complicated. And once I listened to her, I got it. So thank you so much for saving my bum, as it were. And I am so happy to have you here. So thank you so much. And we all look forward to your presentation.
Thank you, Carol. That was a very kind introduction. And I'm really delighted to be able to give this talk today. And you'll have to forgive me if I mess up with the Zoom. While I've hosted a lot of Zoom seminars, this is my first Zoom seminar that I'm giving. I do want to make one small correction. I actually recently moved to the Cleveland Clinic from Case Western. And,
I knew that. I'm so sorry.
Yeah, that's fine. I just, in case there's somebody from the clinic, I have to make sure that I stay true to my new institution. It's actually just 1 mile down the road, and I have retained my position as associate director for basic research in the Case Comprehensive Cancer Center, because the Cancer Center is a multi-institutional center within Cleveland. All right, so without further ado, I'll move on to my talk. By way of introduction, and I probably don't need to do this for this audience, but I go over U.S. breast cancer statistics. In 2018, there were about 250,000 new invasive breast cancer cases diagnosed. The good news is we're reaching cures for almost 80% of breast cancers, but about 41,000 deaths were recorded in 2018. So this means that we still don't know how to treat about 20% of patients. And so my lab is very interested in aggressive disease and what we can do to reduce that number of deaths. In the US, the lifetime risk of breast cancer is about 1 in 8. And lest men think that they're immune to this disease, they're not. In the US in 2018, there were about 2,500 cases of male breast cancer. There are many subtypes of breast cancer, and major part of my lab focuses on triple negative breast cancer, and that's what I'll focus this talk on today, although we do have work going on in ER positive and HER2 positive breast cancer as well. So triple negative breast cancer is how it is defined. It lacks estrogen, progesterone, and HER2 receptors. It is about 10 to 20% of all breast cancers. There are many subtypes of triple negative breast cancer. It's a very heterogeneous disease. Generally, I will call it TNBC or triple negative breast cancer, unless I'm talking about something very specific, and then I might call disease basal if it had been defined by a gene expression profile. This disease is more prevalent in young and African American women, and it is associated with poor outcomes. And this is in part because there are limited targeted therapies for this disease. The only ones that are FDA approved are PARP inhibitors and immunotherapy. So my lab several years ago started looking at gene expression profiles and asking questions about how genes are differentially expressed in different subtypes, and maybe that could tell us a little bit about what was driving their growth. So what I have here is just a heat map showing our favorite genes that are over here on the left. I will talk about the ones in yellow today. And the way these genes are organized in this heat map is by tumors that are estrogen receptor positive or negative. So the negatives are the red, and this was done by immunostaining. And then also by ERBB2 or HER2. And so what you see here is there are many genes that are expressed highly in ER negative tumors, and other genes that are expressed highly in ER positive tumors. So there's a discordance between the different types of breast cancer and what genes they express. And many of these, like I said, are of interest to us because they're expressed in TNBC. So this is a thought experiment, just looking at gene expression, but what we really want to do is understand function, mechanisms, and whether these genes really are important for growth and aggressiveness of tumors. So what we do is once we identify a gene, and genes that we're particularly interested in are those that regulate signaling and transcription. And what we generally do then is see if they associate with outcomes, if they might have an impact on mammary gland development. We do a lot of cell culture studies and look at cell phenotypes. We do many mouse studies all the way from transgenics to PDXs. And then we also do therapy studies. So the first topic I'm going to tell you about, I'm actually going to tell you two stories. One is about bromodomain and extraterminal proteins, and the second one will be about CDK7 proteins. And both of those regulate transcription, so they regulate large swaths of genes within tumors. So the BET proteins, there are 4 family members, BRD2, 3, 4, and a testis-specific one, which is actually not really testis-specific. The most known about the BRD proteins is BRD4. These proteins bind to acetylated lysines, which are readers of the histone code. BRD4 itself is amplified or overexpressed in about 17% of basal breast cancers. And inhibitors of the BET proteins have been reported to disrupt super enhancers at oncogenes and cell identity genes. And I'll get into what super enhancers are in a second. And there have been preclinical mouse studies, quite a few of them actually now, and they had shown that there was limited toxicity. And a variety of these inhibitors have entered early phase clinical trials, and they are associated with thrombocytopenia and neutropenia. And it's becoming very clear that if BET inhibitors ultimately get used for broad types of cancer, that they're likely going to be needed to be used in combination with other drugs. Okay, So for BET inhibitors, there had been this postulate that they may be particularly useful in cancer and that they had low levels of toxicity. And in fact, in mice, they do have very low toxicity. And this is a model that was presented by Rick Young on why BET inhibitors may be more selective in cancer. So for a typical gene, and I just have BRD4 here, but you could substitute any BET protein, it binds to acetylated histones, and this will cause recruitment of other factors and activate a expression of your gene. Now, when you put on a BET inhibitor, you'll lose some of the binding of BRD4 and you'll reduce the gene expression. Now, for oncogenes, BET inhibitors, or BET proteins, sorry, are accumulated in large amounts at oncogenes. And the reason is these genes have what are called super enhancers. So super enhancers are a tandem array of enhancers. There's a large number of transcription factors that bind. There's a big cloud of BET proteins that come in. And so you have this change actually in even in the phase of the aqueous solution around the gene. And this will cause great activation of gene expression. However, when you add a BET inhibitor, now because of all this cooperativity between these proteins, you lose a lot of the gene expression. The expression collapses because the super enhancer collapses. So this is the idea about why BET inhibitors might be more selective in cancer. However, we have a different idea, and I'll explain that as we go through. So when we first got interested in BET proteins and BET inhibitors, our first notion was, let's see if these actually work in breast cancer. So we did some studies looking at breast cancer xenograft models. And what I'm showing you here is a % change in tumor volume for each tumor. So each one of these bars is a different tumor. And we have vehicle treated, Paclitaxel treated as a comparator, and treatment with JQ1, which is a prototype BET inhibitor. So this was a standard xenograft. Here's another xenograft model, and here we see that JQ1 is actually much more efficacious in this model. We've also did this with PDX models, and in each case, we found that the BET inhibitors were effective in inhibiting tumor growth and in some cases causing tumor regression. So this said, yes, BET inhibitors might be a thing in triple negative breast cancer. We wanted to know how they might work. So when I talk to my students, a lot of times I'll talk to them about their therapy studies in cell lines or even with a siRNA or whatever. And I'll always ask them, did you look at your cells? And, you know, you really have to look at your cells. So this is an example where looking at them told us the story. So here are 3 different cell lines, and this is vehicle-treated cells. Blue staining is for the nucleus, for DNA, and we're doing red staining here, it's phalloidin, so we can just see the cell shape. Now, the cells look very different depending on what cell line you're using. But now let's treat them with the BET inhibitor JQ1. What we noticed, and if you can see it here in the insight, is that many of the cells became multinucleated. So this started to tell us that there might be some sort of problem in these cells with either mitosis, cytokinesis, or something like that. We started to think that maybe they might undergo mitotic catastrophe in response to a BET inhibitor. So what is mitotic catastrophe? This is when cells die either in or shortly after mitosis, or they become polyploid and senesce. And we had lots of in vitro work showing that the cells did senesce, and they, as I just showed you, they were polyploid. Now, mitotic catastrophe is a key mechanism of action for certain drugs that disrupt mitotic spindles. An example of this is taxanes, and I'll return to this in a little bit later in the talk. Cancer cells are more susceptible to mitotic catastrophe than normal cells because they have a hard time maintaining their genomes in the first place. So it's thought to be an oncosuppressive mechanism. So how do you determine if your cells are really going through a mitotic catastrophe? Well, one of the ways you can do this is by doing live cell imaging. and simply watching them go through mitosis. Now the time up here, or the numbers up here, is the time that we're watching them go through mitosis. And usually a cell line will have a pretty tight range of how long it takes them to get through mitosis. So in this cell line, if we look at vehicle-treated cells, it takes about 70 minutes for the cells to go through mitosis. Here you can see is the metaphase plate. Hopefully you can see that. Now, when we treat with the BET inhibitor, let's see what happens. Well, now it takes much longer, almost twice in this particular example, for the cells to get through mitosis. Here's another cell line and the same thing. This cell line takes about 120 minutes, but now you're taking about 180 minutes to get through mitosis. Now, a delay in mitosis is a bad thing. You don't want to be in a vulnerable period where you have all of your chromosomes condensed because it's easy for them to break. So being longer in mitosis tells us there's really something wrong with the cells. The other thing we can do is follow what happens to the cells as they go through mitosis. So these are pie charts showing two different cell lines and what happens to them. So in the blue are cells that exit mitosis, and then we wait a while and then they divide again. That's what cancer cells do. Now, as I mentioned before, cancer cells do have some problems with mitosis and maintaining their genomes. So a certain set of them will exit mitosis and die, or even die right in mitosis. Now, this is vehicle-treated cells. This is what happens when you treat with a BET inhibitor. In this cell line, we had a majority of the cells exit mitosis and then immediately die, and then also a large percentage of them died in mitosis. In this cell line, we had a lot of cells exit mitosis and die, many more with treatment versus not treated. But we also had a lot of cells that went into what we call prolonged interphase. So that means as long as we watch the cells, they never divided again, which is probably the early precursors of senescence. And we actually found, which I'm not going to show you any data today, that the difference between these two cell fates is whether you have high levels of expression of BCL-XL. All right, so we wanted to know, how are the BET inhibitors regulating mitosis? And the way we attacked this was we did gene expression profiling. And these are two cell lines where we did the profiling, and this is a volcano plot. And the first thing you should notice with the plot is there are many more repressed genes than activated genes. And then all of these red dots are genes that are known to control mitosis. So this suggested that the BET inhibitors are regulating a large swath of mitotic genes. And some of particular interest to us are shown here, and we confirmed these by real-time PCR, that they were indeed repressed with BET inhibitor. We also did gene set enrichment analysis and found that cell cycle genes that control G2M and MG1 transitions were downregulated in both cell lines. All right, so we know we have a problem with mitosis, and we know we are suppressing mitosis-associated genes. So the next question is, how does this happen? Do BET proteins regulate all of the mitosis genes individually, or might they regulate a core set of genes that then regulate all the other mitosis genes? Well, when we went in and looked at the gene expression data, what we found is that there was a large number of mitosis-regulating transcription factors that were actually repressed. And I'm showing 5 of them here. And a negative control transcription factor is LIN37, where it wasn't regulated by BET inhibitor. So this means that not all transcription factors are repressed by JQ1 or the BET inhibition. We also look to see if BRD4 binding might be changed at these transcription factor genes. And in fact, when you treat with a BET inhibitor, you do see downregulation of binding of BRD4 to those genes. Now, there's a lot known about many of these genes, FOXM1, E2Fs, MIB-L2 is actually BMIB, and their role in breast cancer. But there wasn't really much known about LIN9 in breast cancer, so we decided to take a further look at that. And I went into CBioPortal one day to see whether LIN9 might be dysregulated in breast cancer. And this, to tell you the truth, knocked my socks off. So this is the LIN9 gene. Each one of these rectangles is the expression pattern in a gene. If it's outlined in pink, that's high, it's elevated expression. If it's filled in red, that means the gene is amplified. So for 107 basal-like tumors, and remember basal-like is a subclass of TNBC, 66% of them had overexpression of LIN9. And down here is just showing the heat map of that expression. If you look across subtypes, LIN9 is most highly expressed in the basal subtype of breast cancer. And I started to think, well, maybe this just is because basal-like breast cancers are highly proliferative, and maybe any cancer that's highly proliferative would have a high expression of LIN9. However, if you go in and look at all of these different tumor types, these are some really bad actor cancers. What we found is that only basal-like breast cancer had a very high population of tumors that were amplified and or overexpressed. Other breast cancers, if you look at the whole group, about 30%. And if you look at tumors like lung adenocarcinoma or melanoma, which we know are very aggressive, they had a pretty low percentage of those tumors have high expression of LIN9. So this suggests that LIN9 might have a particular function in basal-like breast cancer. We then also went in and looked at outcomes of patients if they had high or low LIN9 expression. And if you have high expression, you have worse recurrence-free survival and worse overall survival. We had to do this only with all breast cancers because LIN9 is so frequently overexpressed in the basal subtype that we couldn't get a high enough N to be able to discriminate between groups. All right, so LIN9 regulates the cell cycle, and I'll tell you about how that works in a second. First off, I want to remind you about E2Fs. Now, E2Fs regulate the transition through the restriction point in the cell cycle and allow you to get to move towards S. And this E2Fs are inhibited by the RB protein, which we all know is a tumor suppressor. How do you get through the rest of the cell cycle? Well, the way you do that is with a protein complex called MOV-B. And MOV-B is comprised of LIN9, LIN37, 52, 54, and RBB4. MOV-B is actually very interesting because not only does it promote progression through the other phases of the cell cycle, it can also cause entry into quiescence or senescence by interacting with E2F4 and an RB-like protein P130. So LIN9 is the core scaffolding protein for MOV-B, and it is important for progression through the rest of the phases of the cell cycle. And you might recognize some of these other proteins, FOXM1 and BMIB or MIB-L2, which we also showed were repressed by the BET inhibitors. So does LIN9 actually mediate some of the effects of the BET inhibitors? Well, the first thing we wanted to see is if we reduce the expression of LIN9, sort of mimicking what the BET inhibitor was doing, would we phenocopy the BET inhibition? And in fact, we do, and I'll show you a few slides of that. Here, we're just looking at multinucleation. If you suppress LIN9 expression here, and this is showing on the Western that we did indeed suppress expression, you see multinucleated cells. Here's with the JQ1, also multinucleated cells, and actually, surprisingly, it's about the same percentage. LIN9 also inhibits growth, or loss of LIN9 inhibits growth. So the siLIN9 is here in the blue, and the control is in the red. And this is one cell line, and here are two additional cell lines. So when you knock LIN9 down, you reduce the growth of TNBC cells. We also did live cell imaging with SI-LIN9 and found that mitosis took a lot longer. And this is just showing one cell line, but we did this with multiple cell lines. If you silence LIN9, again, you have problems with going through mitosis, and you have a prolonged interphase in this particular cell line. All right, so we can phenocopy BET inhibitors, but does that really suggest that LIN9 is a key target of BET inhibitors? Well, the way we address this is through a computational approach. What we did here is we asked, of all the genes in breast cancer that are correlated with LIN9, so you might suppose that they would be repressed by, or I'm sorry, stimulated by LIN9, of all those genes, which ones are regulated by BET inhibitors? So these are genes that are positively correlated with LIN9, and these are ones that have a low level of correlation. And then what we're looking at is the fold change when you treat with a BET inhibitor. And what we found is if you're highly correlated with LIN9 in breast cancer, you're more likely to be inhibited by a BET inhibitor. Now, if you recall, I told you that LIN9 is amplified in TNBC. So then we decided to break this group down to those genes that were on the same locus as LIN9 versus genes that were on other chromosomal regions. And what we found is even if you're not on 1q, which is where LIN9 is, you're more likely to be repressed by a BET inhibitor than if you are not correlated with LIN9, or even if you're not on the same chromosome. We did another experiment where we looked at, or actually thought experiment, a computational analysis, where we looked at LIN9 binding from a ChIP-seq study. Now this ChIP-seq study was done in HeLa cells, but it was the only study that was available. And what we asked here is, if you are bound by LIN9, are you more likely to be regulated by BET inhibitor than not? And that's actually true. So genes that bind LIN9, about 30% of those genes are repressed when you treat with JQ1. Whereas if you're not bound by LIN9, only about 7% of those genes are repressed by a BET inhibitor. All right, so now we are thinking that LIN9 is an important mediator of BET inhibitor response. The next question we asked is whether LIN9 might regulate response to taxanes. And you might ask, why would we ask that question? Well, when we silenced LIN9, and I didn't show you those data, we saw changes in many of the same cell cycle regulators as we had seen with the BET inhibitors. And I'm just showing three here: cyclin B1, KIF20A, and Polo-like kinase 1. And of course, we had upregulation of cell cycle repressors. Okay, so why am I showing you these three proteins? Well, these guys are all known to regulate taxane efficacy. And so since LIN9 repressed them, we thought, well, maybe LIN9 also will regulate taxane efficacy. So what are taxanes? I think you all know that they're the widely used chemotherapies. It's a microtubule stabilizing agent. They induce multipolar spindles, and it's really a mainstay of therapy for triple-negative breast cancer. And these cancers are highly sensitive to these drugs, but about 50% of patients will develop resistance to them. So there's really a critical need to improve taxane efficacy in TNBC. Okay, so what's the evidence that LIN9 might regulate taxane response? Well, I showed you that it regulated those genes, but more importantly, it correlates with paclitaxel response. So if we look at just a set of 9 different breast cancer cell lines, we assessed their IC50 to paclitaxel, and we found that it was pretty linearly correlated. If you had higher LIN9, you were more resistant to paclitaxel. We also made paclitaxel resistant cells, and these cells were also cross-resistant to docetaxel. And what we found is in the resistant cells that they upregulate LIN9 mRNA, and they also upregulate LIN9 protein. So let's get to the question. Does LIN9 regulate Taxol sensitivity? So what I'm showing you here, this is a complicated graph. It's a dose response curve for Paclitaxel, looking at cell number. And if I can just get you to focus first on the dark red and the dark blue lines. So this dark red line is our parental cell line, and then the dark blue are the resistant cells that we generated through a long-term exposures to increasing doses of Paclitaxel. Now, what happens when you silence LIN9? If you look at the dark red compared to the pink, that's the parental cells. It does repress growth a little bit, or makes them more sensitive to Taxol a little bit. But if you look at the light blue, what you see is you've completely restored taxane sensitivity of the resistant cells. So that's using a genetic approach. I'm sorry, this is another cell line where we see the same thing. And then we decided to use an intrinsically resistant cell line, which is BT549. In these cells, also, if you reduce the expression of LIN9, you restore, or actually, I'm sorry, increase taxane sensitivity. So can LIN9 be therapeutically targeted to improve taxane sensitivity? Now recall that LIN9 expression is repressed when you treat with a BET inhibitor. So we decided to treat the cells with the JQ1 and see if it would also restore sensitivity. So we have the same layout here. Here's the sensitive cells, and we see our Paclitaxel dose response. Here's with the resistant cells, and you see that they, it's shifted the Taxol response curve to the right. And then if we treat with JQ1, we see that we take this dark blue curve to the light blue curve. We've actually sensitized those cells now to Paclitaxel by treating them with BET inhibitor. This is looking at what happens on a cellular level, looking at micronuclei and apoptotic cells. And what I think you can see is that the combination of Paclitaxel and the BET inhibitor greatly increases the cells with micronuclei, telling us that they are having a real problem with regulating their genomic stability, and a big increase in the number of apoptotic cells as well. All right, so you can talk about using BET inhibitors with taxanes, and maybe that would be a useful approach clinically. But it's a BET inhibitors are broad spectrum inhibitors. And we thought maybe there might be a more selective way to inhibit the signaling pathway for LIN9 and still improve taxane response. So how do we find out how to do that? Well, we thought we'd focus on downstream factors of LIN9. Remember, LIN9 is a transcription factor, so it should be regulating the expression of many genes, and some of those might be druggable. Some of the proteins might be druggable. So let's look at the cell biology again. All right, so one of the things we looked at was the centrosomes in cells that had been treated with SI LIN9, so knocking down LIN9. And what we found is instead of having one or two centrosomes, which is the normal state of affairs in cells, when you lose LIN9, you start to get multiple centrosomes. So this cell has 3, and this cell has some sort of centrosome party going on. There's just a whole bunch of centrosomes. And here's a quantitation of that. If you lose LIN9, you have much more abnormal centrosomes than the parental cells. So this is telling us a little bit about, in addition to just the watching chromosomes move mitotic progression, that the centrosomes are specifically dysregulated when you lose LIN9. So then we wanted to find downstream candidates of LIN9 that could possibly be druggable. And we did this again computationally. So what we did is we looked at genes that would be repressed by BET inhibitors, genes that we knew were bound by LIN9 in HeLa cells, but still, at least in that cell line, bound by LIN9, and genes that correlated with LIN9 in breast cancer. And when we overlap all of those sets of genes, we came up with a pretty small set, I think it was 10 genes, that met all those criteria. And then the next criteria we layered on to that was which one of those genes were known to regulate centrosomes. And there was only one gene in that list, and that gene is called NEC2. So NEC2 is NIMA-Related Kinase 2. It's a serine-threonine protein kinase. It regulates mitosis, and it's important for the centrosome cycle. So when you replicate your centrosome, what happens is you will duplicate the centrosome, and then it has to come apart, and one of them has to go to the other side of the cell so that you can form a bipolar spindle. The NEC2 regulates that centrosome disjunction. Is NEC2 really regulated by LIN9? Well, here is a correlation plot in breast cancer looking at NEC2 mRNA versus LIN9 mRNA, and it's a pretty good correlation. If we silence LIN9, we see that NEC2 levels go down. And if we look at our paclitaxel resistant cells, this is 2 different cell lines, we see that in the resistant cells, we have an upregulation of NEC2. And recall, that's very similar to what we saw with LIN9. NEC2 is also associated with worse patient outcomes. And this is looking at all breast cancer subtypes. And then we decided to look at a publicly available data set to see if NEC2 might be associated with response to taxanes. So this data set was published in JAMA, and what they did here is they took patients who were treated with a taxane and anthracycline. They took a biopsy before treatment, and then they followed response of the patients. Now, if you go in and look at those patients, if you look at triple negative breast cancer only, and you look at those patients who had pathological complete response, NEC2 is not associated with relapse-free survival at all. However, if you look at those patients who had residual disease after being treated with a taxane and an anthracycline, what we found now is that NEC2 high expression is associated with worse recurrence-free survival. This is suggesting that NEC2 is regulating chemotherapeutic response, but we can't discriminate between taxanes and anthracyclines. But it does give you some evidence that NEC2 may be important for taxane response. Okay, so the next thing we wanted to do is see if NEC2 might cause similar phenotypes as when you lose LIN9. So here's our silencing of NEC2, and what we find is that indeed they behave similarly to when you silence LIN9. So you get an increase in the duration of mitosis, you have shifts in mitotic cell fates, and this is a cool picture because it actually shows a nucleoplasmic bridge between 2 cells. So this is saying that there's a piece of DNA that's being tugged between 2 cells when you lose NEC2 expression. All right, so there are other phenotypes that we looked at in cells. We looked at the number of centrosomes, if there were some centrosome abnormalities. We looked at micronuclei, multiple nucleated cells, and cells with dysmorphic nuclei. And these are nuclei that are all sort of blebbed and not the typical oval or round shape. And what we did is we looked at if you treat cells with DMSO or paclitaxel, and we compared knocking down LIN9 to knocking down NEC2 to the non-silencing control. And what we found was pretty remarkable. When you knock down NEC2, you get almost identical numbers of these phenotypic abnormalities as when you lose LIN9. And that's true in cells that are treated with vehicle or cells that are treated with paclitaxel. So that's parental cells. We also did this with the resistant cells, and we saw the same thing. Now, resistant cells have much higher levels of these abnormalities because they're trying to figure out a way to get around the taxane inhibition of their microtubules. And what was really notable is when you knocked down LIN9 or NEC2 in the presence of paclitaxel, the cells, almost all of them had these gross abnormalities, centrosomal defects, micronuclei, multinucleated cells, and dysmorphic nuclei. All right, so let's get to the real question. Does targeting NEC2 restore paclitaxel response? And I'm showing you two different dose response curves here for paclitaxel. Again, the dark red is the parental cells, the dark blue is the resistant cells. And when we knock down NEC2 expression with siRNA, that changes the dark blue line to the light blue line. You can barely see it here because it's right over top of the sensitive cells. That's with an siRNA to NEC2. We also used a NEC2 inhibitor. In this case, we used INH1, and we saw a similar thing, that the INH1 brings those cells back to being very sensitive to paclitaxel. We then wanted to look at this in vivo. And what we did here is xenograft models. And as I showed you before, each one of these lines is a different tumor. And this is the % change in tumor volume over the experimental time course. So here's vehicle-treated cells, paclitaxel-treated, INH-treated, which is the NEC2 inhibitor. So paclitaxel and the NEC2 inhibitor have some effect, but when you do the combination, they're much more efficacious. So that's in one model, and this is using a different NEC2 inhibitor, CMP3A, and we see the same thing in another model. And these are both sensitive models that we had seen in vitro were sensitive to paclitaxel. It was pretty interesting here with the CMP3A that the combination actually caused a little bit of tumor regression. We then looked in a resistant model to see if the combination was also more efficacious in that model, and in fact it is. In this case, in the resistant model, they're resistant to taxanes, they're resistant to the NEC2 inhibitor, but the combination is pretty effective. And then we looked at just proliferation in the different models, and we found that the combination inhibits proliferation much more so than either taxane or NEC2 inhibitor. So the conclusions for this part of the talk, with BET inhibitors, they inhibit triple-negative breast cancer growth by inducing multinucleation and mitotic catastrophe. We think that this works in part by repressing LIN9. I think LIN9 may be a novel oncogenic driver in TNBC, and we have yet to figure that out. If you noticed, LIN9 expression was only about twofold elevated, and what we tried to do is overexpress LIN9 in non-transformed cells. And of course, it's very difficult to do a twofold increase in expression, and what we found is that the overexpression killed the cells. So we're still working on that. We found that NEC2 is a downstream effector of LIN9, and what I mentioned is that BET inhibitors synergize with BCL-XL inhibitors. We also found for taxanes that elevated expression of LIN9 and NEC2 are associated with the acquisition of taxane resistance, and that you can therapeutically target these to increase taxane response. All right, I'm going to shift gears and just do a brief vignette on another protein called CDK7. So as we were doing these studies, I started to think about maybe triple-negative breast cancer cells, because they have such problems with maintaining their genomes, they're very genomically unstable, that maybe multiple epigenetic therapies might induce genomic instability in breast cancer. And the reason well, the team had started thinking that is because when we started looking at other epigenetic inhibitors and found that they were all causing similar phenotypes in triple-negative breast cancer. And I'll show you that in a little more detail now. Okay, so we went with CDK7 inhibitors. I'll tell you about CDK7 in a minute. The inhibitors have been shown to be effective in preclinical models of breast cancer. They dismantle super enhancers, which again was how it was proposed that BET inhibitors work. And what I forgot to tell you is that LIN9 and all of those mitosis-regulating transcription factors, none of those have super enhancers. So at least for regulating mitotic progression, the dismantling of super enhancers is not how BET inhibitors work. Okay, so inhibitors were also proposed to act this way. They're in phase 1 and 2 clinical trials. There's a tool compound which we use called THC1. And here's the kicker. In small cell lung cancer, it has been shown that those inhibitors induce genomic instability. So what does CDK7 do? It's a multifunctional kinase. So it's a master regulator of the cell cycle by regulating CDK4/6 and CDK2. It's part of a heterotrimeric complex with MAT1 and cyclin H. It also has another role. It regulates transcription. And it does this by phosphorylating the tail of RNA Pol2. So CDK7 might seem to be a great target for cancer because it has multiple roles that we know are important for cancer progression. In basal-like breast cancer, CDK7 high expression is associated with worse outcomes. I think this is RFS. Okay, so do CDK7 inhibitors work in triple-negative breast cancer cells? Well, yes, they inhibit cell growth. I seem to have lost my name. Oh, here they are. This is 3 different cell lines. If you look at cell cycle progression, we find that the cells get blocked up in G2M. And this is irreversible, the inhibition, because if you do a colony formation assay with no drug present after an initial treatment, you get fewer colonies. Okay, so we did live cell imaging just like I had shown you before. And if you treat with a CDK7 inhibitor, you have a much longer time in mitosis. You also have mitotic fates, like dying in mitosis, exiting mitosis, and then dying, very similar to what you would see with a BET inhibitor. We also saw a lot of nuclear hallmarks of chromosomal instability. So here's some micronuclei. Here's one of those dysmorphic nuclei I was telling you about, and here's a multinucleated cell. And in all cases, when you treat with the CDK7 inhibitor, you increase the percentage of those phenotypes. Now this is pretty cool. This is a confocal image, live cell imaging of cells that are expressing H2B GFP. So that H2B GFP labels chromatin. So if you at normal or non-treated cells, and I have an arrow pointing to this one, you can see it goes through mitosis. So you saw those chromosomes come apart. Now here's what happens when you treat with CDK7. Do you see this cell? Here's a cell and here's a cell, and see this green line? That is a chromosome that's being pulled between these 2 cells. There's another one down here. These 2 cells are playing tug of war with this chromosome. So this tells you that the cells, when they're treated with a CDK7 inhibitor, really have a difficult time with appropriately segregating their DNA. And this cell up here, if you watch it long enough, you'll see that eventually one of the cells wins, the piece of DNA breaks, and it gets the DNA. So how often does that happen? If we look just at mitosis in general, it's very hard to see an actual mitotic event. We see what's happening after that event. And here's the number of, or percent of bridge events, and most of the cells have this bridge event if they look like they've been through a mitosis. All right, so what else happens with CDK7 inhibition? We get double-strand DNA breaks, and I think you would expect that, having seen what those chromosomes look like on the last slide. Using gene expression profiling, we wanted to see who might be the main regulators of CDK7 function. And what we did is did a gene ontology enrichment study using RNA-seq from 2 different cell lines. And the top one here is chromosome segregation. And chromosome segregation, if you look at the list in there, there's a lot of genes that are regulate condensin and cohesin. And those genes are shown here. Most of them are downregulated. And if we look at the core subunits of condensin and cohesin, SMC1A and SMC2, we see that they are indeed downregulated. So condensin and cohesin regulate the structure of chromosomes. Cohesin tethers sister chromatids, and it's really important for transcription. Condensin, on the other hand, is for compaction of chromosomes during mitosis, and it may also regulate superenhancers. And in the interest of time, I'm just going to quickly show you that if you treat with CDK7 inhibitor, you change chromatin states. And if you knock down SMC1A or 2, you can phenocopy the effects of CDK7 inhibitors. And SMC1A, which is the core a core subunit of cohesin, and SMC2, a core subunit of condensin, they both are associated with worse outcomes in basal-like breast cancer. So this second set of conclusions, CDK7 inhibitors induce mitotic catastrophe and genomic instability in triple-negative breast cancer, fairly similarly to what happens with BET inhibitors. So I think this means that the enhancer landscape or regulation of enhancers is really important for maintaining your chromosomal structure, your genomic integrity, and I can get into why that is in a minute or in the questions. Okay, and I didn't have time to talk to you about resistance, but we have resistant cells and we think we know why they develop resistance. So with that, I'll conclude and show you my lab. This is the group of people at CASE, and now they all, well, everybody who's still in my lab moved with me to the Cleveland Clinic. The person who was driving the BET inhibitor studies was Jennifer Sani. The individual who drove the studies with the NEC2 is Melissa Shively. Melissa is here. This is Jenny. And Brian Webb was responsible for the CDK7 work. And then most importantly, Kristen Weber-Bonk is our mouse rancher, and she does all of our mouse studies, and we really couldn't do any of those without her. Collaborators at CASE, pathology support at the clinic, and Matt Summers, who taught me everything I know about mitosis, but I don't know a lot. Okay, I'm happy to take any questions.
Thank you. Wow. I think while we're waiting for some questions to come in, Scott's got a question here, but I just want to ask one real quick question is, so really interesting about the LIN9 regulation. So we also found that LIN9 is regulated very similarly by progesterone in the fallopian tube models that we've been working in. And when we treat with progestins or progesterone, LIN9 goes down like a knockout. And you get cells are inhibited, you get the prolonged interphase phenotype, you get the multinucleate cells, and you get all of that. And so, but interestingly, what happens is those cells do not die. Instead, they become highly invasive, highly migratory. They form spheres. And so if you're not going to kill those cells, they might crawl away. And so the question is, have you done any of that with tumor spheres or stem cells? Or there, I mean, these things go through collagen. And so that was a surprise to us, is that progesterone's protecting, of course, from the proliferative phenotype, but it's causing an invasive phenotype.
Yeah, so we did notice, at least with BET inhibitors, not with LIN9, that the cells did seem to have morphological changes. And we always thought that they might be becoming more stemmy, and but we didn't follow it up. And then there was a publication on that fairly recently showing that there was a shift in stemness with the BET inhibitors.
Oh, that was Ilana's question. It looks like she wanted to know what happens to the stem cells. Okay.
Yeah.
So Scott Dame has a question.
Wait, I'm not done. I want to, yeah. So whether the cells die or not, that's at least in breast cancer cells, or at least in triple negative, it seems to be really dependent on whether you have BCL-XL or not. If you have high levels of BCL-XL, the cells will survive. So I would suggest you look in your cells and see if that's also true, or maybe it's high BCL too. And what we found is if we co-treat with a BCL-XL inhibitor and a BET inhibitor, that they were synergistic and would cause cell death. So that's the way to overcome that problem.
That's interesting because BCL2 is a progesterone receptor gene. So maybe that's why they're senescing.
Yeah.
And not going all the way to death.
Yeah, and you know, senescence isn't really a terminal thing. I think you can come out of that.
Absolutely.
Yeah.
Okay. So Scott wants to know, have you tested whether overexpression of LIN9 or NEC2 in triple negative breast cancer cell line models is sufficient to drive Paclitaxel and or BET inhibitor resistance?
Yeah, we would really love to do that. And we've been asked that a lot of times. And the problem is, and which I tried to explain, is that if you look at the resistant cells, and even in the breast cancer studies, in patients, the elevation of LIN9 and NEC2 is only about 2 to 3-fold. And if you, we tried overexpressing either one, and the problem is it's really hard to get that low of an overexpression. And so what happened is our cells would die. We tried this many different ways. We tried to do an inducible approach. Many ways. And we just haven't been able to get a 2-fold increase in expression. The next thing we're thinking about doing is like a DCAS9 type of approach where maybe there we can tune it a little bit. I would love to know that, but we haven't been able to do it.
That is a tough problem.
Anytime you
Aurora kinase was regulated, what happens to Aurora kinase when you play around with the levels?
Yeah, so if I could just expand a little bit further, any of these cell cycle regulating kinases, they have the same effect if you overexpress or you knock them down, because you're dysregulating their function in the cell cycle. So you can't overexpress Aurora kinases or polo-like kinases either. They all cause cell death. Yeah, sorry.
No, that's right. I agree. We're trying the same things. We see Aurora kinase regulation in our breast cancer models too, and it's very connected with stemness.
Yeah.
Any other questions for Dr. Keri? I don't see anything in the queue.
So I think Dr. Yee has a raised hand, and we can allow him to.
Oh, I can't see anyone. Okay, sorry.
Hi, Ruth, it's Doug. That was great. As you know, triple negative breast cancer has been divided up into multiple phenotypes, largely based on gene expression profiling. So does the LIN9 genotype fall into any specific one? And a question that follows up to a little bit to Carol's. So estrogen receptor positive breast cancers will respond to taxanes, as do HER2 positive breast cancers in combination with HER2 targeted therapy. So have you looked in those types of breast cancer?
I'll answer the second one first. We have not looked in those. It's been reported that BET inhibitors work differently in, at least in ER positive breast cancers where they regulate estrogen receptor function. So they may or may not work there. I haven't tried it, but I'm thinking they may not, because I think triple negative breast cancers have a harder time managing their genomes. They're much more genomically unstable. So I don't know, but I'm guessing they may not work the same way. And then the first question is subtypes of TNBC. So the only thing we've done is looked at either in cell lines, different subtypes, or in breast cancer only at patient samples only at basal. So in basal-like, LIN9 and NEC2 are highly upregulated, and I mean highly. When I say that, I mean percentage-wise. They're upregulated and they're linearly correlated. In the cell lines, it seems like most of the cell lines we've worked with respond to the BET inhibitors by regulating LIN9 and NEC2. So in the cell lines, it doesn't seem to be subtype specific. I don't know, because I'm not sure we can get enough of the subtypes of human patient samples for like the mesenchymal type or LAR type, if the LIN9 would be associated with outcomes or anything, because it's small size.
Okay, we're talking a little bit later. I'll follow up with you on that question.
Okay.
I think we could potentially help with that.
Okay, that'd be great.
Okay, I can't see everyone. Are there other questions? I don't see anything else in the Q&A queue. Nick, do you see anyone?
There was a follow-up, I believe, from Dr. Shvetz. What happens to Aurora A in your first project? Or did we already address?
So BET inhibitors do repress Aurora A and B, and we actually published a paper on that before we got into the LIN9 story. If you reduce Aurora A or B, you get a phenocopy of what you get with the BET inhibitors.
That suggests you would have fewer stem cells, in my opinion. I don't know if Alana would agree. Very cool work. Extremely, extremely exciting. Thank you so much. And
thank you. Thanks for having me.
Those of you are meeting later with Dr. Keri. Thank you for spending time with her, and have a great day. And see you soon, Ruth.
All right. Bye-bye.
Bye.